Great.
This is actually the cutest one that I have. I have a couple of little West Niles that have little Egyptian eyes that are very cool. But you have to be really careful when you say, oh, I have a pathogen, or my friend gave me this disease, when you mean, my friend gave me this cute little stuffed virus. Okay, I'm Dr. Kyle Johnson. I'm an associate professor in biological sciences here at UTEP, and my lab studies how viruses reproduce their genetic material. So a virus, much like this one, are composed of a protein shell, and then inside that protein shell is nucleic acid. It can be DNA or RNA. And this is where the genome is in a virus. And so there are all sorts of complex mechanisms by which the virus has to interact with a host cell, inject its nucleic acid into that host cell, and then proceed to take the cell over. And that really is the basis of human disease, but also gives us the opportunity to study the basic science by which some of these processes occur. And so my lab studies the mechanism by which that genome replication occurs. And a lot of it involves cell biology and looking at how cells and viruses interact. And those virus-host interactions are absolutely critical for understanding how viruses work, and ultimately, if we know how viruses work better, then we can develop new treatments and therapeutics and even vaccines to help prevent them. So we, in the laboratory, study a model virus that is a virus that does not infect humans, and so that's really nice. It gives us a nice, safe model system of a virus that replicates its genomes to such high levels that you can actually see it on a gel and be able to then investigate how that virus is interacting with host cells. We're really doing it within the context of forensic science. And so using forensic science as a model, you could say that we have the objective of looking for the culprit in the virus of hijacking cellular processes, so we can look at viral proteins and cellular proteins and actually see where they occur in the cell, in what quantities, and actually using some really fun techniques, we can see what other proteins they're interacting with. Students involved in this unit will actually be able to publish their results, hopefully, in a scientific journal at the end of the semester. And that's really the goal of this particular course. And so overall, it's going to give the students a flavor for what forensic science is like in terms of tracking down a culprit who's doing a crime, in this case hijacking. Being able to study these model viruses allows us a chance to understand a little bit more about human disease and what we can do with it, because we understand more of those mechanisms by which the virus is able to interact with the host cell and really take it over.